In order to use the information in the DNA, we have to take the DNA molecule and make a small copy of RNA. That's the process of transcription. Once we have that RNA molecule, the next step is going to be taking it and translating it or converting it into a polypeptide sequence. That's going to be the process of translation. Well, the process of translation specifically is using RNA to synthesize a polypeptide, which is going to be composed of amino acids. So it's a large polymer made up of amino acids joined together using peptide bonds. Well, the instructions are going to be in the RNA molecule, and then we will have ribosomes that are able to interpret those instructions. So the ribosomes, they interpret the nucleotide sequence of the RNA molecule. They convert that sequence that they see into an amino acid sequence. The way that they do that is using the genetic code. So the genetic code is what we see right here. This is essentially the language of the cell. This is the language that pretty much all organisms that we know of use. And by language, what we mean is that this is what those letters in the DNA, the bases in the DNA and the bases later in the RNA, this is what they mean. They are going to be read as three-letter combinations, and those three-letter combinations will tell the ribosome what amino acids it needs to be using. So these three-letter combinations that you see right here, that's just one of them. That's called the codon. The codon is used to specify an amino acid. So this is an amino acid. So in other words, if the ribosome sees this combination in the RNA, U, then it knows, okay, in that place I'm supposed to take phenylalanine, which is an amino acid, and that would be the amino acid that I put. Then it's going to look at the next three-letter combination, and it will determine what amino acid that stands for. So the ribosome knows what all of these different codons stand for. There are ultimately 64 of these, so 64 different three-letter combinations, and one of them is what we call the start codon. So this will be at the beginning of all of the polypeptides that it makes. And then we have three that are going to be found at the end of all the polypeptides that are made. So you can see those right here. We have UAA, UAG, and then UGA. All of those mean stop. So again, that would be at the end of the process. So if we talk about participants in translation, we have mentioned a couple of them already. So just to emphasize again, the first participant is that we have to have the instructions. That's the mRNA molecule. So this is the instructions. It contains the codons. This is how we're going to make that polypeptide. It will later be the ribosome that's going to read those instructions and take the information in the codons and translate that, interpret that to mean a specific amino acid sequence. Here we have a look at the DNA sequence. So the DNA is either in the nucleoid or it's in the nucleus. That DNA sequence is converted into an RNA molecule, and then portions of that RNA molecule will contain codons that the ribosome needs to be able to read through later. And so you can see here that it's possible to have an RNA molecule that has multiple regions that have instructions to make polypeptides. So for this particular one, it actually has three different regions that will make three different polypeptides. Each one of them starts with a start codon and ends with a stop codon. It's very common when we're talking about prokaryotes that you have RNA molecules like this, where you have a single RNA molecule that's going to be used to make multiple polypeptides. So RNA molecules, mRNA molecules, those are the instructions. Another participant is going to be a type of RNA molecule that we call transfer RNA or tRNA. The transfer RNA is, again, it's made up of RNA. That RNA creates a unique three-dimensional structure, which you can see in the images here. And its job is to transfer or deliver the correct amino acid to the ribosome. So it will be collecting amino acids and basically passing those off or handing them off to the ribosome. To do this, it will have a three-letter or three-base combination, which we refer to as the anticodon. And the anticodon is going to be complementary or opposite to a particular codon on the mRNA. So in that way, you will have a specific matching with hydrogen bonding between the tRNA and its anticodon and the mRNA and its codon. The ribosome is going to be our main enzyme complex. This is going to coordinate everything. It actually reads the mRNA and interprets the codons, and then it's going to connect the proper amino acids together using peptide bonds. So these are our three main participants that we do have. If we look a little bit closer at the ribosome itself is made up of two subunits. We have what we call a large and a small subunit because they do have different sizes to them. And this is a look at the prokaryotic one. That's the size of it. The S is a sedimentation unit, so it has to do with the size. If we were to look at the ribosome in eukaryotes, it looks really similar, but the size of the subunits is a little bit different. So you can see that the overall eukaryotic ribosome has a larger size. All right, so how is it going to work? Well, the way that it works is that when the ribosome gets ready to actually read through the RNA molecule and make polypeptide, those two subunits, they are going to assemble together into the structure that you see here. And actually, the RNA is going to be threaded right down the center of those two. Now, when we put it all together, within the ribosome itself, there's going to be 3 active sites. And you can see those labeled here. They are EPA, or I like to say APE, because APE really shows you the order that they're going to function in. So notice that in each one of these sites, there are 3 little points that come up into the site, and that's because each site is going to accommodate or fit one codon inside of it. So one codon will be there, and that's on the RNA molecule, and then those 3 can be bound by a tRNA's anticodon. So we're going to see that match be made. Basically, the A site is going to be where new tRNAs come in. So this is where they enter. The P site is going to be where the peptide bond is formed. And then the E site is going to stand for exit. So we will see that tRNAs actually leave when they get to the E site. So if we look at the whole process, this process is going to have initiation, elongation, termination, just like we had when we were looking at the transcription process. So first thing is that this RNA molecule, which has been sent to the ribosome, so basically it's now there in the cell, in the cytoplasm. First thing is that the small subunit of the ribosome has to bind to it. So we can see that happening right here. There are going to be specific sequences there on the RNA molecule that help with the selection of where to actually bind, so where we actually want to start this process. The Shine-Dalgarno sequence is one of those sequences that helps the cell to know this is where we want to bind, this is where we want to start. So it binds in that spot, the small subunit does, and then the very first tRNA, and by the very first tRNA, we're talking about the one that has the anticodon that matches the start codon. When they match up, you can see right here we get very specific hydrogen bonding. So they are hydrogen bonding to each other. And notice that it's got an amino acid attached up here on the top. So that's an amino acid. That's the first amino acid in this new polypeptide that we're making. And once we've got the small subunit bound to the RNA, and then we've got the first tRNA on there, then the big subunit of the ribosome is going to come and assemble and sit on top. And that right there, all assembled together, is what we call the initiation complex. And that is basically the ribosome ready now to get started, ready to go down the RNA and interpret all the information and make the polypeptide from the sequence. If we look at how it's going to happen, basically in the A site, you're going to have a codon, and the next tRNA, whichever one happens to match that particular sequence, is going to come in and bind. And it's going to bring with it the appropriate amino acid. As soon as the amino acid is brought in, look here at how the 2 amino acids get joined together by a peptide bond. The ribosome itself catalyzes the formation of that peptide bond. And then after the bond is formed, the whole ribosome is going to shift over. And as it shifts over, notice that we now have an empty A site, and we have a tRNA that does not have an amino acid on it that happens to be in the E site. So the one in the E site can leave because, again, it's done its job. It has delivered the amino acid to the ribosome. And then we have a place in the A site where a new tRNA can actually come in. So that's essentially what's going to happen. We're going to continue this process with a new tRNA coming into the A site, peptide bond is formed, ribosome shifts over. At that point, the tRNA can leave from the E site, and we've got room for another one to come into the A site. So we'll continue over and over until we get to a stop codon. And when we get to a stop codon in the A site, it will terminate and it will finish up. So termination occurs when a stop codon enters the A site. Okay, and at that point, basically the ribosome lets go, and we've got the polypeptide that's just been made. You've got the RNA molecule, which might actually get bound and read by another ribosome, and then the ribosome is also going to go and find some other molecule to actually work on.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay, so that was prokaryotes. If we contrast it with eukaryotes, they're pretty similar processes. Just a couple things to mention. Initiation in eukaryotes is going to occur when the small subunit binds to the 5' cap. So the 5' cap was one of those post-transcriptional processing steps that we had. Special modification that gets added to the 5' end. So we have the ribosome binding where the cap is at rather than binding to that Shine-Delgarno sequence. The Shine-Delgarno sequence is specific to the prokaryotes. Another thing that's different is that the first amino acid is going to be methionine. So this is a different amino acid. Also, ribosomes, if they happen to be on the rough ER, and we would only find those in the eukaryotic cells, When they're actually making polypeptides, they take it and they thread it right through the ER itself so that they're inserting their new polypeptides into the very interior compartment of the ER, which is what we call the lumen. All right, so those are the differences between translation in eukaryotes and prokaryotes. And when we finish it, we've got a polypeptide that can then be folded up into a very specific three-dimensional shape, and hopefully it's going to be able to perform some type of job inside the cell.